So as you guys have explored NCBI more, so what is NCBI SRA? SRA is called Sequence Read Archive. You have see it's a repository to hold a large amount of sequencing data across the world. Where the National Center of Biotechnology information. It can get information about whole genome data, RNA-seq data, metagenomic and Polygon, or all the different types of data they can hold. So why it is important? What is the main purpose? It is we can able to do reproduce the data where it is publicly available. We can able to create a pipeline. We can do a benchmark to have work with a larger amount of leaders, which is all available. I hope you have explored much about all those things, right? So I just want to add a basic thing over here. I have added. So NCBIs have a understand of big amount of data structures. They have the algorithms of their own where they have been related with GEO. GEO is called then we have SRA, ERA, GRA, and they can have information of bioprojects, biosamples, all specific information, experiment data information, everything, the whole entire public databases they hold. That is where the NCBI. Without NCBI, bioinformatics is nothing in the entire world. So, what is selecting of a microbial data? So it can be of any preference you can have. Why? What happened? My machine is shared only. Is my screen is visible to everyone or not? Okay, I think issue with. Okay, good. Thank you. So the sequencing platform, it is preferred with now I'm going to be more easily we can able to choose with Illumina. Illumina is the core basic on the strong methodology platform it has. And next step is we need to do our sequencing. It's called what is paired end sequencing. Morely we'll use paired end sequencing. And we'll check about the coverage. Coverage, it should be at least 30x coverage, gene coverage data. And if we want to have a complete metadata information, it can have, and we can able to use it for analysis. The specific organism is open to you. You can use any organism, not only a bacterial data. It can be E. coli, Salmo, Staphylococcus, Mycobacterium. Everything will be that. You can able to see. If we can pick any data and then you can able to perform do the analysis. Okay. Just an example of if you want to recreate the same process, you can take any of these organisms and you can able to perform those. Today we'll go with we can able to go with E. coli data, microbial data. So the E. coli is specifically has their specific characters. So the characters it's maybe of gram-negative bacterium, which is the genome of size 5 MB, and the GC count is 50%. Why we have choosing or myself choosing E. coli is it's well studied, which is highly having the high quality reference genomes to perform the analysis. It can hold a larger amount of public data sets which are already available, and which is very easy to start beginner friendly quality check analysis or doing quality performance, all those things. That's why I chose as E. coli. That's not an issue. There are two type of thing. Yesterday you have seen, do you have worked with the web version of NCBI, or you have worked on a command line also, or you have worked on both? That is awesome. You have worked on both. So when a condition is web version, you can able to visualize the data. But as we more of working with the Windows machines, you can't able to download a larger amount of data in the web versions. So while we are working like a larger amount of data, we need to go with the command line based data or FTP to download the data, because the data size is high. You can able to handle it much better. And to do a bioinformatics or NGS analysis, as you guys all know already, Linux is more important, because handling a larger amount of data needs a larger amount of spaces, programming, codings. So for that, more than Windows, Linux is more feasible to perform any kind of bioinformatics analysis. But today, I'm going to teach you about web version only. But if you are handling almost 50 plus data or even 10 plus data, you need to download Linux version, and you can be able to do performance analysis with that. If you are going to perform with Linux version, you need to download the data, and you can able to perform Linux commands like how to cd pattern or grep to down the count, all those things. It is highlighted over here. That's it. One is having a read identifiers, information about the particular sequence of what we are going to perform. And the second line, which is helps to do information about the sequences. And the third line is a separator. And the fourth line is a fast quality score. This is how the FASTQ file looks. And it is the standard format. It is a never-ending format. You need to keep your mind aside that this is the perfect most thing. FASTQ file, so it is going into a more detailed version, the sequences of the DNA sequences, ATCG. Then you can able to see the qualities, that is, which is III. This is all called as ASCII characters. And each character tells these coding values. It's called thread score. And we can able to identify the probabilities of how the errors or things. Each sequences, that is each nucleotides present in the particular FASTQ file, holds the each quality score value. Do you guys know it? Quality score. You have seen the FASTQ file already, right? So how it does the raw data? We are going to work the most important thing. We'll see theoretically for another 30 minutes, and then we'll work on hands-on and in last end if possible. Going into a FRET score. FRET score is also called the quality score. Good. It's the probability of correctness of faces. Yes. So this is the formula that to do calculate the FRET score. Q is equal to minus 10 log P. Q is the quality score. P is the probability of errors, as you guys are said. To understand the quality values, here is a table which just tells you our qualities score value, which is started with 10, 20, 30, 40, 50s and all. It is based upon the error rate, which is telling if it is at 10, the probability that the number of bases present 1 among 10, and then the accuracy is 90%. If it is 20, that is then the error values of bases is 1 among 100, that is 99.9% of accuracy. Then there's 30, that is the error in 1 among 1,000, which is 99.9% accuracy. Then 40, 50, 12 goes on. What is the standard quality of data we consider to perform the next step of analysis? Which level we need to fix it? You have know it. What quality score will consider to perform the next set of analysis? It's already in my slide. What standard score we consider to perform the analysis. What stand with the value which is below will reject. It's already in my slide. It's Q30. At least it should be Q30. The quality score should be Q30. Where the error rate, the accuracy should be 99.9%. Then only we can able to do perform the analysis. That is where we consider those data. The qualities which is below 30 Q30 we don't consider because there where the error rate will be high, we can't able to have a better results to perform the analysis. This Q30 is right. Yes, that's what I have mentioned over here. The common quality thresholds. The Q30, which is most acceptable, which is above Q30, which is to do an analysis, to do a pre-processing, to understand other types of variant calling, the end of ingest analysis, downstream analysis, all can be taken after that. Another metrics which is important to improve the quality is called quality rate, per million opportunities, that is DP, percentage of process capacity. This is all will be comes under doing any types of other metrics we can able to consider when we are working with the gene expression data or any data or any other different types of source of data. In that case, it will be important over that. And coming into a different quality matters is the quality content will be focused, will be deferred in different applications. Why that does matters? It is important to do a genome assembly process. It is also important to do a variant calling. If the quality there may be a false or a bad SNP detection is possibly we can able to identify. In taxonomical mistakes also will be taken place. It will increase the compatibility, computational cost. If we do a low quality data, it increases the processing time. This is a waste of time, right? So we need to be a proper data to handle to do analysis. So quality matters more. What determines the quality of a sequence? Sequencing methodology, sequencing platform, or the data quality, everything will be comes under that. We'll see what are the determines matter. That's what we are going to see in the quality sessions. So